Okay, so this video will hopefully help you work through our experiment on spectroscopy. And so what I have, you should be able to see my screen. And I have three tabs open, the first of which is the actual experiment Google Doc. And then we'll go through the other two as we kind of get through it. So the idea of this Google Doc, if you look at it, we've got some overall background on spectroscopy, the interaction of light and matter. So you should read through this, some explanations. If we keep scrolling down, we'll eventually get to the procedure. Because this is a remote lab, what you want to do is kind of read through the procedure, and then there's embedded videos that you can watch just so you get a sense of what would happen if you were actually in the lab. So what I would do is encourage you to open this link, this first link, read these instructions. This is about calibrating our SpectraViz, and then see how it's done. And we're going to do that through each of the parts. So part A, part B, and then part C. If we keep scrolling down, I'll come back up in a second. I just want to show you, here's the data sheet. So what you want to probably do is save yourself a copy of this Google Doc. And so you can have one that's editable. And when you have one that's editable, you can kind of be filling in the data sheet as you're working through the procedure. So we'll scroll back up. You can see the first thing on the data sheet would be part B. So let's go back up to the procedure for part B. In part B, what we're really starting to do is investigate this relationship between the color of a solution that we see and the absorption spectrum. And so what you'll do is you will look at and collect data for a red, yellow, green, blue, and purple solution. And so to do that, you can see the procedure shown here of how this was done. And what you can do is you can click on this video that shows the data collection. And so here's that video. I'll back it up so you can see there's no sound on the video, but there is some text to sort of highlight things. And so we're going to, oops, I think that's actually the wrong video. So let me back up to the right video. There it is. Investigation of color, absorbance and color. So you can kind of go through this and you can see here's the red solution. It goes into the SpectraViz. We can see data collection here. And then you can kind of see the spectrum of what it looks like here. So then this gets stopped for a second. I'm going to pause the video for a second, because if we go back to the Google Doc, what you see is this folder, this link to a Google Drive folder. And so in this folder, if I go to this folder, you can see what the full spectrum of all of those solutions will look like. So for that red solution, we just looked like, if I double click on it, it's going to open. And you should be able to download this if you want. This is just a picture file, or you can just open it in the folder. Either one would work. And what I want to sort of point out to you, I'll keep this tab open and go back to my data sheet now. And I'm going to scroll down to that first table. So in part B, for the red solution, we want to know what were the colors that were absorbed, what were the colors that were transmitted. So that means what light went through. And then what is the lambda max? What is the wavelength with the highest absorbance? So let's come back to that picture. Here is our, remember, this solution was red, and the y-axis is absorbance, and the x-axis is wavelength. And for our red solution, what we see from our plot is that solution does not absorb red. In fact, it transmits red because there's no absorbance. That means all of the red light goes through. And in fact, the reason we see this as being red is the red light goes right through. And so we see red. But all of the other colors, you can see yellow, green, blue, and purple are absorbed, right? And so when we go to this table, we would say what colors are absorbed. We might write yellow, green, blue, purple are absorbed. What colors are transmitted? Red. And then for the wavelengths of maximum absorption, what we're looking for is an estimate of the peak heights. In this case, I have two peaks. This one here in the purple, this one looks pretty close to 400 nanometers. And I have a second peak here, right in the green. And this is going to be, I don't know, come down here. This is an estimate, maybe 520 nanometers. So for this first box, we could write the colors absorbed, transmitted, and for the wavelengths of maximum absorption, we might write 400 nanometers and 520 nanometers. We can then do the same thing for the green, the blue, and the purple, and you can find those files here. And again, we're just trying to do the same analysis that we did with the red, but now with these solutions. So that'll take you through this first table. Once you have this filled out, we can start to answer question 2. Because what I hope is you'll start to see a pattern of what the color we perceive of the solution and how that relates to these two columns, color absorbed versus color transmitted. And that's the explanation we're looking for here. In question 3, it's going to ask you to apply this knowledge. And so what you want to do is be able to predict what would the spectrum of an orange solution look like. So if our solution was orange, can you sketch what we'd see for an absorption? What colors do you think would be absorbed and what colors do you think would be transmitted if our solution had an orange color? And we can kind of draw this. If it's hard for you to draw on it, you can sort of type in text to explain what you'd see. Either one's fine. All right, so that takes us to part C. And so for part C, I'm going to come back to the procedure. Here's part C. So again, here's the instructions. So you should read through these, and then as you do so, we can watch this YouTube video. And this is the investigation of absorption and concentration. And in this case, we're dealing with a yellow solution, right? And what we're going to do is we start off with a solution that's pretty yellow, and we're going to dilute it and measure it and dilute it and measure it. And so each of these solutions will be a little bit less yellow. All right, so when we come back, oops, let's go back here. So here we have our procedure. You're going to watch this, and then for our analysis, in the end, here are your 3 solutions. Right, so we did 3 solutions, and the absorption values are absorption of 1.522, 0.887, and 0.478. So these values are what go in this table. The most concentrated was the stock, and then solution A, and then solution B. Okay, so we can read these right off the plot. The lambda max, again, is an estimation of where that peak occurs. And this is going to be the same for all 3 of these because it's the same color, right? These are all the same molecule makes the yellow color. So it's going to have the same lambda max. So you can estimate it off the plot and fill in the table. For the concentration of solutions, we have to calculate these. And we have 2 calculations to do. All right. And in order to calculate the concentrations, we're going to use our equation. If I scroll back up, you can see in our introduction, we're going to use this C1V1 equals C2V2. And there's a sample calculation here. This is our dilution. I think we also sometimes call that M1V1 equals M2V2, where the concentration molarity times the volume equals the concentration times the volume. All right, so in this case, C1 is going to be your concentrated solution, so 1 molar. V1 is how much of that did you take out. V2 is the final volume, and we're solving for C2. And if you solve for C2, that'll give you the solution A, concentration. To find solution B, concentration, it's basically the same idea, except C1 is what you just calculated for solution A, because we're doing a serial dilution. If you have questions on that, please let me know. So finally, what we want to do is we want to make a plot. And in order to make this plot, what we need to know are these 2 columns. And we're going to make a plot of concentration versus absorbance. And so you can see that in the procedure. Actually, that's down below. Sorry if it's scrolling. In number 8, this link will help you walk through this and how to make this plot. But again, in order to do that, we need these 2 columns full. All right, and so there's 4 data points that will go on that plot. Your concentration is the x-axis, your absorbance is the y-axis. All right, and from that, you can make your plot. All right. If you have questions on any of that, please let me know. I hope that helped. Please let me know if there's more questions. Thank you.